So, good morning, everybody. Is the volume okay? Okay, so thank you for the introduction. Today I will speak about part of my research job as a PhD student, which consists of a repeated sorption process of TBP on wood and its removal by Trametes versicolor through solid-state fermentation. So first I would like to talk to you about organophosphate flame retardants. These are compounds that are used as additives in many products such as furnitures, plastic, foams, etc. And the problem here is that as they are not chemically bounded to their host material, they tend to diffuse into water compartments. So as you can see here in this graph, this is a study that they did in 5 different wastewater treatment plants in Catalonia about the efficiency of these plants to degrade this kind of contaminants. And today I want to focus on Tributyl phosphate, or TBP, which is a flame retardant that has been detected in 100% of the wastewater effluents of these plants. And there's also a similar behavior in other research performed in other countries. So it's only partially removed in the plants, and the danger is that it can be really toxic for animals, plants, and for the human beings. So one really appealing alternative is the use of white rod fungi, which are fungi that are able to grow on wood, so are therefore able to degrade a complex substance such as lignin through a very non-specific extracellular enzymatic system and a very versatile intracellular enzymatic system, which is the cytochrome P450. So given this combination of enzymes involved, they have proven to be able to degrade a lot of emerging contaminants, and that's why we plan to use them. So what we did first was to test a fungus that our group has been working on for years, which is Trametes versicolor. And we tested first the degradation of 10 ppm of TBP in a liquid culture media. And we saw that the contaminant was completely degraded after 7 days. So the next step was to set up a reactor, and then we planned that maybe it would be a good idea to use the wood as a support for the fungus. So what would be the advantages of this? First, that of course the wood can work as a selective carbon source for the white rod fungi, so that way we are enhancing the growth of the fungus. But it also can prevent bacteria competition because, of course, we would like to work under non-sterile conditions. However, there is a big but, which is the contaminant sorption because of the porosity of the wood. So basically, our purpose of this work was to evaluate if is the fungal able to degrade TBP when this is sorbed into the wood support. So for this, first we wanted to study how the TBP sorption on oak wood after different saturation cycles to see if the wood can get to an equilibrium where it doesn't absorb any more contaminant. Then we wanted to find out if the contaminant can inhibit the fungus growth after the inoculation. Third, we wanted to follow TBP's degradation in contaminated wood after the inoculation. And finally, to identify transformation products that would allow us to propose a degradation pathway. So this is the first part of the experiment, which are the saturation tests. So what we did was to take autoclave wood, oak wood, and we would put it in contact with a TBP solution for 2 days. Then we would separate the phases, and we would measure the concentration in the liquid phase to determine how much of the contaminant was absorbed into the wood. Then we would take a sample that we would use afterwards for the inoculation of the fungus, and we would continue to saturate the wood, like repeating these cycles for 10 times, always adding more fresh water to the wood. So for these first sorption experiments, what we expected after many other research works of our group was that the wood would stop absorbing the contaminant after some 5 or 6 cycles, but we actually saw that even after 10 cycles, the wood keeps absorbing the same amount of contaminant, so we have a linear trend. And we actually got to an amount of 0.35 milligrams of TBP per grams of dry wood, which is a really big amount. So after we saw that the wood can absorb a lot of TBP, then we performed an inoculation. And after the inoculation, we took samples after 30 days, 60 days, and 90 days of growth for each of the cycles of the saturated wood. We would treat the samples, and then we would perform an extraction for quantifying the biomass. So the quantification of the biomass, we did it through ergosterol, which is an esterol that the fungus generates in its cell wall for growing. So here at a first glance, you can see in the graph here in the left that it appears that the fungus can grow in a stable form as regardless of the cycle of the saturation, so regardless of the concentration of the TBP on wood, the growth is the same. And this also happened for 60 days and 90 days, so the growth was kind of stable. So then we decided to perform a control of the wood without TBP presence, and that's where we realized that there is actually an inhibition in the growth, even for the first days of the inoculation, because as you can see here in the control, I don't know which. Here in the control, there is actually a difference in the growth already of 25%, and of course this difference it's bigger as the inoculation goes by, because when in the control sample the fungus is growing even more. So actually TBP inhibits the growth of the fungus from the start. So we already know that the contaminant can inhibit the growth of the fungus, but we wanted to see if even the growth was inhibited, the fungus was still able to degrade the contaminant. So for the same samples, we perform a different extraction procedure, and we would determine the TBP. So as you can see here, regardless of the initial concentration of the sample, the fungus is still able to almost fully degrade the contaminant after 60 days of inoculation. And the difference in removal is almost the same between 60 days and 90 days. And also, we calculated a removal rate. And the most important one was between 0 and 30 days, because it's when there is more amount of TBP. And we actually saw that there's also a linear trend in the removal rate. So we believe that the fungus is able to maintain its degrading performance even at high contaminants concentrations. So also for validating the degradation, we wanted to identify transformation products. So we were able to identify 3 different transformation products. And what we did was to check the behavior of the areas between samples taken at 30, 60, and 90 days for the same saturation cycle. So this is the first product that would be expected for the degradation in one of the degradation paths. And as you can see here, there's a big amount of the product at 30 days, but it starts to diminish as the inoculation goes on. So this is really important, because although we saw that at 60 days there was almost a complete degradation, between 60 and 90 days you can see that the degradation pathway can continue to evolve. So we would have more degradation products, which is what we expected. We had the same situation with this other product. And finally, we had this third product, which was kind of like the final product for the degradation that we could identify. So in this case, you can see that the behavior is different, because this product progressively appears as the inoculation performs. So we think that this is kind of like the final stage of the pathway that we could obtain. So based on these results, this is the degradation pathway that we could find. I will not go very much into detail, but pretty much we think that TBP goes first to 2 hydroxylation steps. This is the first one, and this is the second one. And afterwards, for 2 dealkylation steps. We found that this pathway has been also proposed for TBP degradation in mammals, where they found out that the main responsible for this degradation was the intracellular system, which was cytochrome P450. So it's the same intracellular system. So in conclusions, we saw that after 10 cycles of contact with the contaminant at a concentration of 10 ppm, the wood continues to sort TBP without any signal of saturation. Also, we saw that fungal growth is inhibited by the presence of TBP. We plan to do toxicity tests both for the contaminant, also for its transformation products, to kind of elucidate what is going on. And we also saw that although TBP can inhibit its growth, Trametes versicolor is able to still degrade it after 60 days of contact, regardless of the concentrations that we tested. And finally, which is I think the most interesting part, is the degradation products that were identified, because we were able to really verify that the intracellular system has a really important role in the degradation of this compound. And it is also something that we were able to verify in our lab as well. But we still think that the role of the extracellular enzymes should be eluded, so we have to determine it yet. And that's all. Thank you for your attention. I will be happy to respond to any questions.